Hello everyone. Today is the eighth problem solving session for the course Environmental Chemistry and Microbiology. The course instructors, as you know, are Professor Anjali Pal and Professor Sudha Goyal from IIT Kharagpur, and I am Shrija Ghosh, a PMRF from IIT Kanpur. Till now we have discussed topics from environmental chemistry, and then in the previous session we had started the introduction to microbiology. So today we'll continue the microbiology part. Today we'll talk about cell chemistry, different type of cell chemistry, different type of components in a cell, and then we'll talk a little about different type of microscopic techniques as well. Okay, so I've prepared certain questions which are similar to the assignment questions given to you. We'll discuss the questions, and I request all of you to answer those questions. You can write the answers in the chat box, and then we can discuss if your answer is correct or wrong. Other than this, if you have any form of doubt and any other questions apart from the ones that are discussed today, please let me know. I'll try to help you out. Okay. Okay. So I'll start with the questions now. I hope I'm audible to everyone. Is my voice clear?
Yes, ma'am.
Yeah, okay. Then I'll start with the class. So, starting with the first question, question number 1. Bacterial cells can be stained with dash to reveal the presence of lipid inclusions. Bacterial cells can be stained with dash to reveal the presence of lipid inclusions. Options are safranin, methylene blue, trypan blue, and Sudan dyes. Which of the following can be used to stain the lipid inclusions in a bacterial cell? You can write your answers in the chat box. I can see one answer in the chat box already. All of you write your answers. Okay, I can see one answer. Maisha has answered option A. Other than that, all of you have answered option D. Anyone else wants to write their answers? All of you write your answers, even if it is wrong, it's okay. We'll have a discussion. So, okay, I see now more answers in the chat box. Most of them are option D, Sudan dyes. Anyone else wants to answer? Okay. Let's see the correct answer. The correct answer is option D, Sudan dyes. So Sudan dyes have a high affinity towards fats. Therefore, these dyes are used to reveal the presence of lipid inclusions. So lipids are composed of fats and fatty acids. Okay, so Sudan dyes, they can go and bind to those fats and stain the lipid inclusion. So these dyes are mainly used to stain any kind of lipid mound structures. Okay. Other than that, you can see three options: safranin, methylene blue, and trypan blue. Right. So safranin is a pinkish, reddish to pink color dye, and they stain the cells pink. So if I stain the bacterial cells with safranin, the cells will appear pink in color. Methylene blue, from the name itself, you can see it has a bluish color. So methylene blue is a positively charged dye, and because of the positive charge, it can actually stain the negatively charged uh, molecules. So you know that DNA is a negatively charged biomacromolecule. So methylene blue can actually stain DNA and structures that contain DNA. So it can stain the nucleus. Methylene blue is a positively charged dye, and it can stain the negatively charged DNA. Okay. Trypan blue, on the other hand, it is a negatively charged dye, and trypan blue is actually used to check the cell viability. Okay, so how does it actually work? So trypan blue, being a negatively charged dye, so you know this cell membrane is also negatively charged. So whenever we stain a live cell with trypan blue, the live cells won't be able to take up the trypan blue, because the negative charge of the membrane will repel this negatively charged dye. Okay, so what happens, the cells that are dead, since the dead cells start uh, rupturing their membrane, so the membranes of the dead cells start rupturing, and uh, there are gaps in the boundary of the cell, so that allows the trypan blue to enter. So only the dead cells will appear blue in color, and the live cells will have no color. Live cells will appear transparent under the microscope. So the shiny transparent cells are the live cells, and the blue stained cells are the dead cells, when the cells are a mixture of cells are stained with trypan blue. And then we can count the number of dead cells and live cells to determine the viability. Viability means how many live cells are present in the culture. Okay, so all these dyes are actually used for staining, bacterial cell staining. Every dye has its own purpose, has its own property. So Sudan dyes are have a high affinity towards fat. Trypan blue, it's a negatively charged dye. It is used for cell viability determination. Then methylene blue, it is a positively charged dye, so it is used for staining the negatively charged structures. Then safranin, it is actually used in a staining procedure called gram staining. We'll discuss about it in detail in the upcoming slides. Okay. Anybody has any doubt in this? Please let me know if you have any doubt or any questions regarding this. Okay. You can write your doubts in the chat box, or you can unmute and ask. Meanwhile, I'll move on to the next question. Question number 2. Cell size and cell number. Cell size and cell number can be measured using, options are hemocytometer, Coulter counter, Petrov-Hauser counting chamber, and none of these. Cell size and cell number can be measured using, option A hemocytometer, option B Coulter counter, option C Petrov-Hauser counting chamber, and option D none of these. How can we measure the cell size and cell number? Write your answers in the chat box. All of you write your answers. I can see only 3 answers. What about others? Anyone else wants to answer? Okay, the three answers in the chat box are option B, Coulter counter. All three are option B. Okay, I have one more answer from Olivia. Option C, Petrov House accounting chamber. Okay. Anyone else wants to write the answer? So the correct answer is option B, Coulter counter. So three of you have written Coulter counter is the correct answer. So here the all three instruments that are mentioned, hemocytometer, Coulter counter, and Petrov House accounting chamber, they are all used for counting the cell number, determining the cell number. Only Coulter counter can be used to determine the cell number and the cell size. So this is how a Coulter counter works. So this is a diagrammatic representation of Coulter counter. So the cell suspension is passed through this flow cell, and one by one each cell will pass through this cavity, and when this cell is passing through the cavity, this is connected to a circuit, and a current is uh, gen there is a change in the current because of the interference. There's a interference in this current because when the cell is passing through this cavity, it is causing an interference in the current. Okay, and the number of times that interference will be recorded would be the number of cells, whereas the size of the cell can be determined by the intensity of the interference. So if the cell size is bigger, there will be a larger interference, and if the cell size is smaller, there will be a smaller interference. Okay, so the Coulter counter can tell you both the number of cells by counting the number of interference and the size of cell depending on the intensity of the interference. Okay, whereas the other two options, hemocytometer and Petrov Hauser counting chamber, they are used for counting the cell number. So both of them are essentially the same or a similar thing. So it's a slide like this, a glass slide, where two of these units are made. So these parts are edged, and it has a structure like this. So you can see the grid here, right? So when you keep the slide under a microscope, you can see this grid through the microscope. Okay, that this grid is made in this one of these parts. Okay, so there are two. Generally the hemocytometer comes with the two grid formation so that you can load two sample in one slide. So we keep a cover slip over this, and you can see these tiny grooves here, right? So you can use a micropipette to load the sample from this groove. First you place the cover slip and then you load the sample. Usually 10 microliter sample fits in this volume, and then we have to put the slide under the microscope and count the number of cells in these grids. Now for for bacterial cell count, bacterial cells are smaller in size. Okay, for that we use these smaller grids in this Petrov Hauser counting chamber, and for mammalian cells which are bigger in size, we use these larger squares. Even for blood cell counting, the name hemocytometer actually comes from that there, because these slides are also used for counting the blood cells. So I think you all have seen, if you have gotten a blood test ever done, there is a complete blood count report that comes. Okay, so for that, nowadays there are sophisticated machines and automated systems, but then initially the counting used to be done in such kind of uh, using such kind of methods. Okay, so there are two types of grids here. You can see one which are larger squares and one which are much finer squares. Okay, so in the larger square, this one big square where my laser pointer is, this whole thing is 1 mm by 1 mm dimension. Okay, the square is 1 mm by 1 mm, and the volume that holds in this particular uh, grid is zero point 0.1 mm. So basically the height between the cover slip and the slide. So that makes the total volume that is held by this particular grid is 0.1 mm cube. Okay, so now based on the number of cells that we see in this grid and then in this grid here and here, so we can see four similar sides, right? So we'll count the cells in all these four corners and then we'll take an average number of cells. So let's say the average comes to 20, and then we'll multiply it with the uh, we'll divide it with the volume. So we'll get the number of cells per ml or per volume unit, whatever unit volume is there, we'll get the cell count like that. And in the finer grids, we generally use the finer grids for bacterial cells which are much smaller. So here the uh, so here you can see clearly, so this finer grid, so it has smaller, even smaller division. So you can see small squares in this fifth quadrant, you can see, right? So one small grid actually has these many uh, so this whole fifth square, which is 1 mm by 1 mm, this fifth square has 25 tiny squares, you can see here. And in each tiny square, we'll count the number of cells, and then we'll multiply, we'll take the average and we'll divide by the volume. So that is how we count the number of bacterial cell using this chamber. And one small square's dimension is 0.02 mm. The volume is 0.02 mm, height is 0.02 mm, and the dimension is 0.2 mm. Okay, so this is how we determine the cell count. Is this clear to everybody? If anyone is confused, please let me know.
Ma'am, could you please explain the last diagram? I mean, Petrov-Hauser counting chamber.
Yes. So this is basically to show you how do we count the cells. So you can see this fifth square, right? The square that is labeled 5. So the dimension of this whole square is 1 mm. Okay, now this square is further divided into 5 by 5 squares. Okay, so 25 squares, it is divided into 25 squares. So 1 mm divided by 5 would be 0.2 mm. So one square, if I take the dimension of this number 1, okay, that would be 0.2 by 0.2 mm, the dimension. Okay, and the volume that is there inside one tiny square would be 0.02 mm. So your height of the sample, so your total volume would be length into breadth into height. So 0.2 multiplied by 0.2 multiplied by 0.02. Okay, so that would be your total volume in ml or in mm cube. Okay, now why this volume is necessary? Because you want to count the number of cells per ml. Okay, so for that volume is necessary. Now when we load the sample in this slide, when we load our sample, let's say we have a bacterial suspension culture, and we load that culture, we dilute that culture, and then we load that culture. Then under the microscope we can see these cells here in the magnified picture. You can see the number of cells, and then you count the number of cells. So you will count only the cells that are present inside the grid. There will be cells that are present outside the grid, but we won't be counting that. We'll only count the cells which are present inside the grid. And there is a general rule that is followed, we if the cells are on the on the border of the square, then should we count it or should we not? So the general rule that is followed universally is, we generally count the cells if it is touching the border of the left or the top edge. We don't count the cell if it is touching the border of the right or the bottom edge. Okay, so then you count these cells. Okay, so let's say here we have 10 cells, then like that we'll count the number of cells in, let's say, 4 square. Okay, you don't have to count it in all 25 squares, you can count it in 4 squares. Let's say I count it in 1, 5, 25, and 21. Okay, and then I'll take the number of average number of cells basically. So let's say here I had 10, here I had 15, then here also I had 10, here also I had 15. So I'll take the average. So 10 plus 10 plus 15 plus 15 divided by 4. Then whatever will be the average will be, let's say around 12.5. That will be the average number of cells in this particular grid. That average number of cell will be divided by the volume that I told you how to calculate the volume. And based on that, and how much microliter you loaded, based on that you will calculate the initial number of cells per ml in the suspension that you loaded in the slide. Is this clear to you now?
Okay, ma'am. Thank you, ma'am.
Yeah. So this is how cell counter, uh, like when you have to count the cells manually, this is how we do it. So for mammalian cell culture, this is still very commonly used. We all use it in our lab for counting the cells. For bacterial cells, we generally don't use such a cell counter. And nowadays, these automated cell counters are also present, where you can just insert this kind of slide. You can just insert the slide and the machine can read the number of cells and tell you the number of cells. So you don't have to actually like count the cells under the microscope. Machine can tell, count and tell. Okay, so moving on to the next question. If
Ma'am,
any, yeah, tell me.
ma'am, one student is trying to join. Please accept her.
Yes, yes, I've counted. Okay, so moving on to the next question. Most microbial structures and enzymes are composed of, options are, option A lipid, option B proteins, option C carbohydrates, and option D lipids and carbohydrates. Most microbial structures and enzymes are composed of, option A lipids, option B proteins, option C carbohydrates, and option D lipids and carbohydrates. Write your answers in the chat box. Okay, I have 5 answers in the chat box. What about others? Anyone else wants to answer? Okay, all 5 answers are option B, proteins. Let's see the correct answer. The correct answer is option B. Proteins form up to 55% of the dry weight of bacterial cell. Even the virus coat is made up of protein that envelops the nucleus. So, protein is the most abundantly found biomacromolecule in a cell. Uh, in the dry weight of the cell. So percent dry weight percentage holds up 96% of the dry weight, is composed of the macromolecules. So there are different macromolecules present in the cell: proteins, polysaccharides, lipids, lipopolysaccharide, DNA, RNA. From that, 55% major portion of the macromolecule is proteins in the cell. Then comes the RNA, which is 20.5%. Then we have other polysaccharides, we have other macromolecules like polysaccharides, lipids, and lipopolysaccharides. Other than these macromolecules, 96% of macromolecules, we have 3% of the monomers, which is the structural unit of these macromolecules. So for proteins, amino acids and precursors are the uh, monomer units of the protein. So amino acids join together to form the protein. Similarly, sugars join together to form the polysaccharides, and nucleotides join together to form the DNA and RNA. So there are different monomers present that makes these macromolecules. So 3% of the monomers are also present, along with the 96% of macromolecules in the dry weight of the cell. Other than that, 1% is composed of the inorganic ions. Okay. So the major portion of the dry weight of the cell is composed of proteins. Okay. Anybody has any question in this? Please let me know. Moving on to the next question. Which of the following organelles contain DNA, divide, and possess some degree of autonomy? Which of the following organelles contain DNA, divide, and possess some degree of autonomy? Options are: option A, Golgi apparatus; option B, ribosome; option C, mitochondria; and option D, peroxisomes. Which of the following organelles contain DNA, divide, and possess some degree of autonomy? Golgi apparatus, ribosome, mitochondria, peroxisome. Write your answers in the chat box. I can see 4 answers in the chat box. All of them are option C, mitochondria. Anyone else wants to write the answer? Sorry. Anyone else wants to answer? Okay. Okay, let's see the correct answer. So 4 of you are saying option C, mitochondria. That is the correct answer. So mitochondria and in plants chloroplast, these two have evolved from the specialized bacteria which have survived endocytosis by other cells and got incorporated into the cytoplasm of the cell. Hence they are known as semi-autonomous organelles. So these organelles have evolved to become evolved from the bacteria. So our cells, whenever there are bacterial infection, or if there's a surrounding particle present, our cells have the capacity to endocytose those particles and then later kill those bacteria by different mechanisms. Okay, so all the organisms have this ability. uh, some or the other form of mechanism to take up the particles surrounding it. So ages ago, probably there were some cells which have endocytosed some bacterial cell. Instead of killing those bacterial cell, that bacteria somehow survived and evolved to become a organelle inside the cell itself, and then all the cells started to have that organelle inside them, and that organelle was mitochondria, and in case of plants it was chloroplast. why it was believed to be present from like evolved from a bacterial cell? Because it has its own DNA. It possess some level of self division capability, so it is kind of like a live cell inside the living cell. Okay, so inside our cells there is another organelle which in itself looks like a living cell, because it has its own DNA. It can multiply, it can like it can live on its own, so it has some level of autonomous properties. Okay, so that is why that organelle is believed to be evolved from some living cell itself. So it is believed to have evolved from a specialized bacteria after endocytosis, which has survived the endocytosis, which did not die during endocytosis, and it just evolved to become a organelle and permanently stayed there in all the cells that came after it. Okay, any doubt in this? If you have any doubt, please let me know. Okay, meanwhile I'll move on to the next question. Question number 5. Calculate the wavelength of the light if diameter of the smallest resolvable object is 250 nanometer and the specimen is observed with 100x oil immersion lens having numerical aperture of 1.2. Calculate the wavelength of the light if diameter of the smallest resolvable object is 250 nanometer and the specimen is observed with 100x oil immersion lens having numerical aperture of 1.2. If you know the formula, calculate and let me know the answer. Options are 360 nanometer. Okay, there are two 360 nanometer. So you have three options: 360 nanometer, 720 nanometer, and 600 nanometer. Calculate if you know the formula. If you know the formula, calculate and write your answers in the chat box. Anyone is able to solve this? Anybody? Okay, I see two people have solved it. Both of them have given the same answer, option D. What about others? So let's see what is there in the question, what is given in the question. So in the question we have the resolution given as 250 nanometer and the numerical aperture is given as 1.2. So there's a formula which is a relation between the resolution, wavelength and the numerical aperture. So resolution is directly proportional to the wavelength and inversely proportional to the numerical aperture. So resolution is equal to lambda, that is the wavelength, divided by 2 into Na. So or 0.5 lambda by Na. Na is the numerical aperture. So now I've told you the formula, so solve it using the formula and tell me the answer. I see two more people have meanwhile answered in the chat box. What about others? There are almost 15 students in this call and I have only 4 answers. I've told you how to solve it now. So you have the resolution. Resolution is equal to the wavelength, 0.5 wavelength by numerical aperture. So you have the numerical aperture, you have the resolution, calculate the wavelength. It's not a very big calculation. Okay, so the 4 of you who have answered option D, 600 nanometer, that is the correct answer. So this is how we have to solve it. So we have, I've told you the formula, resolution is equal to 0.5 lambda by numerical aperture. So resolution is 250 nanometer. So 250 nanometer is equal to 0.5 lambda by numerical aperture is 1.2. So 250 is equal to 0.5 lambda by 1.2. Now you can solve this. 250 into 1.2 divided by 0.5 or multiplied by 2. So if you solve this, you will get the answer to be 600 nanometer. Is this clear to everyone? Is it clear? Anybody has any doubt? People who haven't answered in the chat box, especially those, do you have any doubt in this? Okay. If you have a doubt, please let me know in the chat box. Meanwhile, I'll move to the next question. Transmission electron microscope is best to observe option A stained bacterial smear, option B unstained live tissue when it is desirable to see some intracellular structure, option C intercellular details of a cell that is 1 micron long, option D a sample that emits the light when illuminated with UV light. Transmission electron microscope is best to observe. What is the purpose of using transmission electron microscope? A stained bacterial smear, unstained live tissue when it is desirable to see some intracellular structure, intercellular details of a cell that is 1 micrometer long, or a sample that emits light when illuminated with ultraviolet light. What would be the correct answer? All of you write your answers in the chat box. I have only one answer in the chat box. All of you should write the answers, then only I'll know if you know the answer or if you are understanding or not. And if you are not understanding something, please ask. Even after I explain, even after that, if you don't understand, please ask again. I don't mind explaining it again and again. But you need to ask in case you have a doubt. Otherwise, I won't know if you have a doubt or not. I see 4 answers in the chat box. All of them are option C. Anyone else wants to answer? Okay, let's see the correct answer. So 4 of you are saying option C, intercellular details of a cell that is 1 micron long. That is the correct answer. So transmission electron microscope, from the name itself you can see it's an electron microscope. So the source of illumination is electrons. So the normal microscope that you must be familiar with has a light bulb. So the source of illumination there is normal visible light, and those microscopes are called optical microscopes. In this case, the source of illumination is electron. So, electron microscope, and transmission stands for whether the electron beam is transmitted through the sample or not. So, there are two types of electron microscope commonly used, TEM and SEM. So, TEM stands for transmission electron microscope, SEM stands for scanning electron microscope. So, if the electron beam transmits through the sample, passes through the sample, so here the sample is here and this is the source of electron gun. So, electrons are passing through the sample. So, this is transmission electron microscope. Whereas, in scanning electron microscope, the sample is here, the electron beam comes, hits the sample and gets reflected from the sample, and those reflected electrons are captured by the detector and the image is framed. Okay. So, there are two types of electron microscope. In the transmission electron microscope, the electrons are passed through the sample, and the scanning electron microscope, the electrons hit the sample and bounce back. Okay. The purpose of transmission electron microscope is to resolve the intercellular structure. So, since the electron beam is transmitting through the sample, it is passing through the sample, it actually makes it easier to visualize some internal structures, because it is passing through the sample, it is passing through the internal structures and it is able to get the images of those internal structures. So, this is a TEM image of a phospholipid bilayer where you can see the intercellular space of the bilayer. If you would have done a SEM on this, scanning electron microscope, you would have only seen the surface of the sample. So, in SEM, it hits the sample and bounces back, and those bounced back electrons are captured by the detector. So, only the surface is scanned and surface can be imaged. Whereas in transmission electron microscope, the internal structure of the sample can be seen. Okay. And you know how in a normal microscope, in an optical microscope, we have lenses attached which focuses the light towards your sample. Similarly, in this case, since the source is electron, the illuminating source is electron, to focus those electrons towards the sample, we have uh, electromagnets attached to the instrument. So, these lens are basically electromagnets that focuses the electron beams towards your sample. Okay. Is this clear? Anybody has any doubt or any questions regarding EM? Let me know. If you have any kind of questions regarding this, let me know. Okay, let's move to the next question if there's no question, if there's no doubt. Match the appearance of gram-negative bacteria after following steps in gram staining. Match the appearance of the gram-negative bacteria after following the steps in gram staining. So, there are different steps of gram staining given on the left hand side and different colors given on the right hand side. You have to match each step and their corresponding color. So, steps are first is crystal violet, second is alcohol acetone, and third is safranin. And the colors given are first is purple, second is pink to red, and third is colorless. So, you have to match the appearance from the steps. So, you have to match the appearance with the steps followed. Write your answers in the chat box. Okay, I can see most of you have answered option C. One person has answered option B. Let's see. So option C is crystal violet 1 1. So crystal violet purple, alcohol acetone colorless, and saffron and pink to red. Yes, so option C is the correct answer. So all of you have answered option C is the correct answer. So I have told you in the first question, saffron has a pinkish color. It gives the cells pink to reddish color. And crystal violet, by the name itself, you can see it has a violet color, so it stains the cells violet or purple. Okay, so what is gram staining, or why do we do gram staining? So there are two types of bacteria present, gram positive and gram negative, and this name is given based on the staining procedure actually. So this I have discussed in the previous class also. So gram positive bacteria have a thick peptidoglycan layer, whereas gram negative bacteria has a thin peptidoglycan layer. So in the gram staining procedure, we start with crystal violet. So first we take the bacterial cell and make a smear on a glass slide. Okay, a thin smear on a glass slide. We heat fix that smear so that the bacteria sticks to the slide and all the cells are killed, but the morphology is restored. So after heat fixing, we stain the cells with crystal violet first. Okay, so we add the crystal violet, keep it for 1 minute, and then wash it off. So cells take up the crystal violet. So be it gram positive or gram negative, all the cells will take up the crystal violet. Okay, the second step is to add the grams iodine, which is a iodine solution, which is added to uh, make the crystal violet bind properly to the cell wall. So be it gram positive or gram negative, now crystal violet is bound to the cell. So if you observe the cells just after addition of crystal violet, you will see the cells are appearing purple in color. Okay, so after addition of crystal violet and grams iodine, the third step is to do the decolorization by using alcohol or acetone solution. So when you add alcohol to the crystal violet stained cells, now what happens? The alcohol washes off the crystal violet. But now the thing is, gram positive bacteria has a thick cell wall, thick peptidoglycan cell wall. Okay, and the gram negative bacteria has a thin layer of peptidoglycan. So when you are washing the crystal violet out of the cells, it is easier for gram negative to lose the crystal violet. For gram positive, the crystal violet doesn't wash off easily. So after decolorization using alcohol, if you observe the gram negative cells, they will appear colorless. But if you observe the gram positive cells, they will still appear purple, because the gram positive cells having a thick peptidoglycan layer, that crystal violet seeps into the thick layer of peptidoglycan and does not leave even after washing with alcohol. Okay, and after the decolorization step, we stain it with a counter stain called saffronin. So saffronin has a pinkish reddish color and it stains the cells pink. So the cells which are now colorless will take up the pink color and they will appear pink under the microscope. The cells which are gram positive cells, which are still purple after the decolorization, they won't be able to take up saffronin and they will keep appearing purple, dark purple in color. So basically after doing all the 4 steps of gram staining, when you observe a mixture of gram positive and gram negative under the microscope, you will see the gram positives to be purple and gram negatives to be pink in color. Okay, and the appearance after each step for a gram negative would be purple after crystal violet, colorless after alcohol step, and pink after saffronin. Okay, so gram negative cell walls have a thin layer of peptidoglycan in the periplasmic space within its inner and outer lipid membrane. Cell wall takes up crystal violet dye, appears purple. Dye and mordant complex made by iodine is easily removed by the uh, alcohol, making the cells colorless, and then this colorless cell wall takes up the counter stain, that is safranin, and appears pink to red. Is this clear to everyone? I hope everybody understood the gram staining procedure. If you have any doubt on this, please let me know. Any doubt? Okay. If there's no doubt, then I can move to the next question. Anybody has, if anybody has a doubt, please write it in the chat box. I'll read it. Okay. Or if you want to unmute and ask, you can do that as well. Meanwhile, I'll move to the next question then, if there's no doubt. Question number 8. Which of the following pairs is mismatched? Which of the following pair is mismatched? Option A, bacterial identification matched with gram staining. Option B, dark field microscopy matched with light passes through the cell. Option C, three-dimensional image, atomic force microscopy. And option D, immersion oil prevents refraction of light and its scattering away from the lens. Which of the following pair is mismatched? Write your answers in the chat box. All of you write your answers. I can see 4 answers in the chat box. All of them are option B. Dark field microscopy, light passes through the cell. Let's see the correct answer. That is the correct answer. So all the other 3 options that are there, they are correctly matched. So bacterial identification, I have already explained how gram staining is used to identify whether a bacteria is a gram positive or a gram negative kind. Then atomic force microscopy, it is a type of microscopy which is used to determine three-dimensional structure. So you can determine the height and the morphology of the structures, morphology of the sample using this type of microscopy. Then comes immersion oil. The use of immersion oil is to prevent the refraction of light and its scattering away from the lens. So that is correct. So we use a drop of immersion oil on the sample when we are using a 100x lens to observe bacterial cells, because at that higher magnification, if the light scatters away, we won't be able to see a higher resolution sample image. So the only mismatched pair here is dark field microscopy and light passes through the cells. So in dark field microscopy, there's no light passing through the cells. In fact, the light gets reflected from the cells or specimen. So it uses a special condenser with an opaque disc that blocks the light from entering the objective directly, and light is reflected by the specimen, enters the objective lens, and the specimen appears light against a black background. So under dark field microscopy, we have a dark background and we can see the specimen. If I have a spherical cell, let's say, so, and if I observe it under dark field microscopy, I'll see a black background and small spheres appearing which are lighter in color or which are transparent in color, like which are transparent, and it will appear against that black background. Okay, so that is dark field microscopy. Anybody has any question, any doubt, any comment? Please let me know if you don't understand any of these things. I'll move on to the next question, question number 9. Which of the following is not accomplished by fixing bacterial cells to a slide? Which of the following is not accomplished by fixing bacterial cells to a slide? Option A, cell morphology is preserved. Option B, cells are killed. Option C, cell structures are differentiated. And option D, cells are attached to the slide. Which of the following is not accomplished by fixing bacterial cell to a slide? Cell morphology are preserved, cells are killed, cell structures are differentiated, and cells are attached to the slide. What would be the correct answer? Write your answers in the chat box. All of you write the answers in the chat box. Okay, I can see 5 of you have answered. Anyone else wants to answer? Anybody? Okay, so 5 of you have answered option C, cell structures are differentiated. That is the correct answer. So heat fixing is done during the gram staining. I've explained that when I was explaining gram staining. So fixing of bacterial cells basically is to adhere the cells. First of all, it is done to adhere the cells to the slide so that it doesn't move when we are observing them. uh, it is also done to kill the cells again so that we can see the cells shape of the cell properly, and obviously the cell morphology, the shape of the cell is preserved when we are doing this heat fixing, because we want to see that particular morphology, so it needs to be preserved. So the process that we follow during the fixing, that makes sure that the cell morphology is preserved, the cells are killed, and they are attached to the slide. The only thing that is not accomplished by fixing this fixing of cells is cells differentiation of cell structure. So you can't differentiate between different cellular structures uh, by cell fixation. Cell fixation, it attaches the cells to the slide so that they can be used for microscopy or any other analysis. It allows most cells morphology to be preserved but does not allow cells to be observed in a living state. Okay. Is this clear to everyone? If you have any doubt, please let me know. Okay. You can ask your questions in the chat box or by unmuting your mic. I'll move on to the next question, question number 10. Suppose you observe that bacteria are moving in your gram stain. You can conclude that option A, the bacteria are gram negative. Option B, the bacteria have capsules. Option C, you did a flagella stain, not a gram stain. And option D, you didn't fix the smear. Suppose you observe the bacteria are moving in your gram stain. You can conclude that. What can you conclude if you see the bacteria moving in the under the microscope? All of you write your answers in the chat box. I see 5 answers. What about others? Okay, so 6 of you have answered. Option D, you didn't fix the smear. That is the correct answer. So fixing of smear, as I've told you, it is done to attach the cells to the slide. So if you don't fix it properly, you will see the bacteria moving because they are not adhered to the slide properly. Okay. I hope this is clear to everyone. If you have any questions, any doubts, please let me know. Meanwhile, I'll move to the next question.
Can you show the previous slide once again?
Yeah, sure. This one?
Yes, ma'am. Thank you.
Okay. Okay, I can move to the next question then. Next question is question number 11. Ability of lenses to distinguish 2 points at a specific distance, at a specified distance apart is termed. Options are magnification, resolution, numerical aperture, none of the above. I can already see answers in the chat box. Okay. So 5 of you have answered already. Option B, resolution. Anyone else wants to answer? Ability of lenses to distinguish 2 points at a specified distance apart is termed as. Okay, so all of you have answered option B, resolution. That is the correct answer. So resolution basically means the ability of a particular lens or particular device to separate two objects, like to distinguish two objects as separate entities. So I'll explain in terms of a normal photograph. Whenever we see a image, we generally say like, oh, the resolution of the image is good, or the resolution of the image is bad. Or even while watching a video or a movie, if it is blurry, we generally say that the resolution is so bad. What do you mean by the resolution in such cases? Basically, if you see the items blurry, let's say it's a picture of two people, but it's a blurry image, so you can't really figure out if there are two people or three people or more. Okay, so it's a blurred image. So that means the photograph that is shown to you, that cannot resolve between the distance of those two people. Okay, so let's say those two people are 2 points. So the camera by which that image is taken, by which that photograph is taken, that camera cannot resolve the distance between those two people, and thus we see a blurred image and we cannot distinguish those two persons as individual entities. Okay, similarly, when we are talking about a microscope, it is also like, it also has a lens, it is also an imaging device at the end. Okay, so like your camera, microscope is also an imaging device. So when we are using a microscope, how well your microscope can see two different points as two separate entities, that is the resolution. So the distance between 2 points, minimum distance between 2 points that can be observed under a microscope would be its resolution. Okay, so it's a function of wavelength used. So we have seen in the numerical question. So resolution is a function of wavelength used and the numerical aperture. So resolution was equal to 0.5 multiplied by the wavelength divided by the numerical aperture. So it depends on both wavelength and the numerical aperture. It is the ability to distinguish 2 points separated by a minimum specific distance. For example, the resolution of most common optical microscopes is 0.2 microns, which means that objects separated by minimum distance of 0.2 micron can be distinguished as two or more objects if the separating distance is less than 0.2 microns. Now, if we have two bacterial cells which are very close to each other with a distance of less than 0.2 microns, then we won't be able to see those two bacterial cells as separate cells. We will see them as one single cell or a blurred kind of image. Okay, if you have two bacterial cells separated by the distance of 0.2 microns, you can see those two cells as separate cells. Okay, so that is what we mean by resolution. Is this clear to everyone? It's a very common term that you must be using in your day-to-day life also, so you should know these things. Like when you say the resolution is good or bad, you should know what actually resolution means for a image, for a photo. So microscopic image is nothing different from a normal camera image. It's just that it has a better resolution than your camera. Your camera can't see the bacteria, microscope can. That's the only difference. Anybody has any doubt? Any questions? I'll move on to the next question then. Match the following macromolecule and prokaryotes with their location. So you have different locations given on the left side and different macromolecules given on the right side. So the locations are cytoplasm, nucleoid, cell wall, and cytoplasmic membrane. The macromolecules are DNA, peptidoglycans, lipids, and proteins. Match the following. Write your answers in the chat box. I see one answer. All of you write the answers. I see 4 answers. All of them are option C. Anyone else wants to answer? Okay, so most of you are saying option C. Let's see. Option C says first matches with 4, cytoplasm for proteins. So proteins are present in cytoplasm. That is correct. 2, 1. So nucleoid and DNA. So DNA is present in nucleoid. So I told you in the previous class the difference between prokaryotes and eukaryotes. Prokaryotes don't have any membrane bound organelles. So the DNA is present in a prokaryotic cell in one location, but it is not surrounded by any membranous structure. And the location where the DNA is present is called nucleoid. In eukaryotic cell, the DNA is present inside the membrane structure called nucleus. In prokaryotic cells, it is present in the cytoplasm, but the region is called nucleoid. Okay, so nucleoid contains DNA, cytoplasm contains protein. What is next? 3 is 2. Cell wall contains peptidoglycan. I explained you in the previous session also and in this session also while explaining gram staining. Peptidoglycan is the main component of cell wall. Then you have 4, is cytoplasmic membrane, which has the lipids. So membrane is composed of lipids. So we have the plasma membrane or the cytoplasmic membrane. These are composed of lipid. Phospholipid bilayer is the membrane's composition. So it is composed of lipids. So option C is the correct answer. Anybody has any doubt or any questions, please let me know. Moving to the next question. Ribose and adenine can be found in. Ribose and adenine can be found in option A, ATP and RNA, option B, proteins and lipids, option C, DNA and RNA, option D, ATP and DNA. Ribose and adenine can be found in option A, ATP and RNA, option B, proteins and lipid, option C, DNA and RNA, option D, ATP and DNA. I can see 3 answers in the chat box already. Write your answers in the chat box, all of you. All of you write your answers in the chat box, then we'll discuss what is the correct answer. Okay, I can see 4 answers in the chat box. All of them are option A, ATP and RNA. That is the correct answer. So ribose and adenine, both of these are found in ATP as well as in RNA. So RNA, the difference between RNA and DNA is the presence of the sugar. So in RNA it is the ribose sugar, and in DNA it is the deoxyribose sugar. So the other options have DNA in it, so it won't be the correct answer, and proteins and lipids don't have either of these. So the correct answer is option A. Adenine is present in ATP. So ATP is called adenosine triphosphate. So adenine is the base. When it is attached to the sugar, it is called adenosine, and 3 phosphate groups are attached, so it becomes adenosine triphosphate. So ATP is a nucleotide that consists of 3 main structures: the nitrogenous base adenine, which is this, then the sugar ribose, and then chain of 3 phosphate groups bound to the ribose. RNA is a ribose nucleic acid and contains ribose as well as adenine along with other nitrogenous bases such as guanine, cytosine and uracil. So RNA is composed of 4 bases, A, G, C and U, whereas in DNA instead of uracil we have thymine. Okay, is this clear? Anyone, if you have any doubt, yeah, tell me. Do you have a doubt, Mobani?
No, ma'am.
Okay.
This is clear, ma'am.
Okay, great. Okay, then I'll move to the next question. True or false? We have 2 statements. You have to tell both the statements if they are true or false. First statement is nucleic acids are found in storage granules. Second statement is proteins are found in cytoplasm, cytoplasmic membrane, cell wall and flagella. So you have to tell if both the statements are true, both the statements are false. First is true, second is false, or first is false, second is true. Write your answers in the chat box. Okay, I can see 3 people have answered option D. That is, first is false and second is true. So first statement is nucleic acids are found in storage granules, and second statement is proteins are found in cytoplasm, cytoplasmic membrane, cell wall, and flagella. Okay, more people have answered. So all of you are answering option D. So you are saying first statement is false and second statement is true. That is the correct answer. So the nucleic acids, they are not found in storage granules. Storage granules generally consists of lipids and polysaccharides. Nucleic acids, they are actually found in nucleus or nucleoids in case of prokaryotes. Proteins, whereas they are found everywhere. They are found everywhere. I've told you 55% of the dry weight of a cell is composed of protein. So they are found in cytoplasm. They are found in cytoplasmic membrane, which are the membrane proteins. Then they are found in cell wall, and they are found in flagella. Okay, so mostly the cell is composed of protein. So they are the building blocks, kind of. Okay. So nucleic acids are found in cytoplasm, nucleoids of microbes as DNA, and in the nucleus and eukaryotes. Storage granules contains carbohydrates or other substrates that may be needed by an organism when the substrate or food in the environment is scarce. So by the name itself, you know that it's called storage granules. It is storing some form of food that can be used up when the cell is starving. Okay. So any doubt in this, please let me know. Meanwhile, let's see the next question. Match the following monomers to their polymers. So you have monomers on the left hand side and the polymers on the right hand side. Match the monomers with the polymers. So the monomers are sugars, amino acids, nucleotides, and fats or fatty acids. And the polymers are nucleic acids, polysaccharides, lipids, and proteins. Match the following. Write your answers in the chat box. There are 3 answers already. All of you write your answers. I can see all of you who have answered, they have answered option C. Let's see that is the correct answer or not. So option C says first is matched with 2, sugars is matched with polysaccharides. That is correct. Polysaccharides are composed of sugars. Sugars are the monomers. And then second is matched with 4, amino acids with protein. That is also correct. Then third is matched with 1, nucleotides, 2 nucleic acids. That is also correct. So nucleotides form the nucleic acids. And fourth is with 3, that is fats or fatty acids. They are the building blocks for lipids. So option C is the correct answer. Anybody has any doubt in this? This is pretty self-explanatory. If you have any questions regarding this or regarding any of the questions discussed till now, please let me know. You can either write it in the chat box or you can unmute your mic and ask, whatever way you feel comfortable. But if you have a doubt, please ask. This is the time to ask. Okay, let's move on to the next question. Question number 16. Which of the following is not a function of nucleotides? Option A, participate in oxidation reduction reaction in the cells. Option B, carry sugars in biosynthesis of polysaccharides. Option C, regulate or inhibit or stimulate activities of certain enzyme. Option D, provide structural integrity to the cells. Which of the following is not a function of nucleotides? Which of the following is not a function of nucleotides? Write your answers in the chat box. I can see 3 answers already in the chat box. All of you write the answers. Others who haven't answered yet, write your answers in the chat box. Okay. 5 of you have answered option D, provide structural integrity to the cells. That is the correct answer. It is not the function of nucleotides. Structural integrity is provided by cell walls. So in mature cells, cellulose and lignin forms the secondary cell wall, and primarily, the cell wall in bacteria is composed of peptidoglycan. The cellulose and lignin cell wall is generally found in plant cells. Okay, so structural integrity to the cell is provided by cell walls. Nucleotides can participate in oxidation reduction reactions. So when we'll study about biochemical pathways in the cell, we'll see the molecules like ATP and GTP, they actively transfer the phosphate group to different molecules. So for example, a process called glycolysis, when it happens, the glucose takes the phosphate from ATP to form glucose 6 phosphate. So that's the oxidation reduction reaction that happens inside the cell, and nucleotides take part in it. Then second option, carry sugars in biosynthesis pathway of polysaccharides. Nucleotides are composed of sugars. So we saw the structure of ATP. ATP had a base, nitrogenous base. It had the sugar and it had 3 phosphates attached, right? So the sugar part of the nucleotide, that is actually provided, and when polysaccharides are biosynthesized. So during the biosynthesis of polysaccharides, the sugar is actually taken from the sugar backbone of the nucleotides. Then the third option, regulate, inhibit or stimulate activities of certain enzymes. Certain enzymes are activated or regulated by the presence and absence of ATP. So ATP or GTP, they come and bind to the cell or to the enzyme or some part of the enzyme, and then the binding actually facilitates the change in the pocket of the enzyme, so that one substrate can fit or maybe one substrate cannot fit, and that's how the enzyme activity is regulated inside the cell by these nucleotides. Is this clear to everyone? If you have a doubt, please ask. The next question is, most abundant substance in cells is options are option A, protein, option B, water, option C, sugar, and option D, none of the above. Most abundant substance in the cell. Which of the following is most abundant substance in the cell? I can see 5 answers in the chat box. All of them are option B, water. Anyone else wants to answer? Okay, let's see the correct answer. The correct answer is option B, water. That is the correct answer. So the cell, actually 70% of the total cell mass is made up of water, and the remaining 30 is that dry weight that we talked about in one of the previous questions. So from that 30% uh, of the total cell mass, which is the dry weight of the cell, in that 55% was composed of protein. But overall, if you see, 70% of the cell is made up of water, and it is the most abundant substance in the cell. Okay, clear? Which of the following, according to the shape and arrangement of bacteria, is correct? Name the following according to the shape and arrangement of bacteria. Options are option A, diplococci and streptobacilli. Option B, first is staphylococci, second is streptobacilli. Option C, first is tetrad, second is cocobacillus, and fourth is option D is first is streptococci, second is diplobacilli. What would be the correct answer? I can see option B in the chat box. Okay. Most of you have answered option B. Okay. Anyone else wants to write the answers? Option B, that is first is staphylococci and second is streptobacilli. That is the correct answer. So here are the different shapes of bacteria that is defined. So basically, if a cell is spherical, it is called coccus or cocci. Cocci is plural and coccus is singular. And if the cell is rod shaped, it is called bacillus. Bacillus is singular and bacilli is plural. So one rod will be called bacillus, multiple rods will be called bacilli. One sphere will be called coccus, multiple will be called cocci. Now, if there are two of them attached together like this, then it will be called diplococci. And two rods attached like this, they will be called diplobacilli. Okay. If they are arranged in long chain, they are called streptococci if sphere and streptobacilli if rod shaped. If they are arranged in an arrangement of four spheres, it is called tetrad. If it is arranged in arrangement of 8 spheres, it is called sarcina. Okay. And if it is forming a bunch of sphere, it is called staphylococci. And for bacilli, if the shape of the cell is rod with rounded edges, it is called cocobacilli. So it is like a cocci and a bacilli, so it is called cocobacillus. Okay, a rod shaped and spherical. Okay, so these are the different shapes of bacteria and different names given to these shapes. Okay, depending on
Ma'am,
the.
could you please show us the previous slide?
Yeah, this is the previous slide. So here you can see this is staphylococci and this is streptobacilli, rod shaped, arranged in chains. So if it is arranged in chain, the term strepto will be there. If it is rod shaped, bacilli. If it is round shaped, it will be cocci. So if it was long chain of sphere, it would have been streptococci. This is rod shaped, so streptobacilli. And this is a bunch of cocci, so it is called staphylococci. Okay. There are other shapes also. Comma shaped is named as Vibrio. So the common cholera bacteria, Vibrio cholera, it is shaped like a comma, like how we make this comma. Those cells are called Vibrio.
Thank you, ma'am.
Okay. Okay. Anybody has any doubt or any questions? Okay, moving on to the next question. Question number 19. The plasma membrane is considered a barrier to the environment because options are option A, it is made up of structural proteins. Option B, it is rigid structure. Option C, no molecule may pass through it. And option D, it controls the passage of molecules into the cells. Plasma membrane is considered a barrier to the environment because. Why does a plasma membrane considered barrier? I can see 4-5 answers in the chat box. All of them are option D. It controls the passage of molecules into the cells. Most of you have answered option D. Let's see, that is the correct answer. So plasma membrane, it is considered a barrier to the environment because it can control the passage. So it is called a semi-permeable membrane. It allows certain molecules while blocking others. So molecules that are needed by the cell, only those molecules are allowed to enter the cell. Rest of the molecules are not allowed to enter the cell. So it is a semi-permeable membrane and they are made of phospholipids. Phospholipids have a amphipathic property. So amphipathic means the one end of the molecule is polar and the other end is non-polar. So in these phospholipids we have a polar head group which is made of glycerol and a non-polar fatty acid chain which is forming like a tail of this phospholipid. So the polar head group and the non-polar for fatty acid chain. So and it's a bilayer membrane. So you can see the in the bilayer the fatty acids are facing towards each other making a hydrophobic core. So this will be non-polar or hydrophobic core and the edges of these membrane will be hydrophilic facing the outside which is uh, mostly water or aqueous. So aqueous can interact with polar and the non-polar will be buried inside. Okay, so they make a impermeable membrane through which only gases can pass. Even water cannot pass through this membrane because water cannot interact with these non-polar fatty acid chain. So for transport of different substances into the cell, there are different proteins embedded in these membrane which helps the transport of different molecules. Okay, so these proteins are the membrane proteins. Is this clear? Okay. Anybody has any questions? Or can I move to the next question then? Mopia, which slide do you want to see? This one only, right?
Yes, ma'am, it's done. Thank you.
Okay. Okay, then moving on to the next question. Active transport is different from facilitated diffusion because active transport requires, so more than one option is correct in this question. Active transport is different from facilitated diffusion because active transport requires, options are option A, osmotic pressure, option B, lipid bilayer, option C, energy in the form of ATP, and option D, a transporter protein in the plasma membrane. Active transport requires osmotic pressure, lipid bilayer, energy in the form of ATP, and a transporter protein in the plasma membrane. What would be the correct answer? Write your answers in the chat box. Remember, more than one option could be correct in this question. So read the question carefully, read the options carefully and answer. I can already see 3 answers in the chat box. Others also write your answers. So the three of you who have answered options C and D. C is energy in the form of ATP and D is transporter protein in the plasma membrane. Let's see the correct answers. The correct answers are option C and D. That is, you guys are right. So 4 people have answered and they have answered correctly. The process of moving molecules across membrane of cells using cellular energy in the form of ATP is called active transport, while facilitated diffusion or facilitated transport is a spontaneous passive transport process of molecules or ions through the membrane mediated by proteins. Okay. Any doubt in this? So option C and D are the correct answers. Anybody has any doubt? Moving on to the next question. Which of the following linkages are present in cellulose? Which of the following linkages are present in cellulose? Options are option A, alpha 1,4 glycosidic bond between D-glucose molecules to form straight chains. Option B, beta 1,4 glycosidic bonds between D-glucose molecules to form straight chains. Option C, alpha 1,6 glycosidic bond between D-glucose molecules to form branches. And option D, both A and B. Which of the following linkages are present in cellulose? I see 4 answers on the chat box. All of them are option B. What about others? Everybody write your answer. Okay. So most of you have answered option B, beta 1,4 glycosidic bond between D-glucose molecules to form straight chain. That is the correct answer. So cellulose is formed by D-glucose molecules. So there are 3 types of glycosidic linkages: alpha 1,4, beta 1,4 and alpha 1,6. So let's see how these uh, linkages are formed. So the difference between alpha and beta glucose is the position of the OH on the first carbon. So if I have to number these carbons, so this will be the first carbon, this will be the second carbon, this will be the third, fourth, fifth and then sixth carbon. Okay. So the position of OH, if it is on the same side on the first and the second carbon, then it is called an alpha glucose. If the position of OH is opposite on the first and the second carbon, it is called beta glucose. Okay. So if two alpha glucose are linked by the first and the fourth carbon, so this is the first carbon in this alpha glucose and this is the fourth carbon in this alpha glucose. If the OH is linked by a glycosidic bond between the first and the fourth carbon of the alpha glucose, it is called alpha 1,4 glycosidic bond. If the bond is between two beta glucose and between the first and the fourth carbon, it is called beta 1,4 glycosidic bond. And in case of two alpha glucose, sometimes the OH of the first carbon can link with the OH of the sixth carbon, that is the carbon outside the ring structure. So that will make a alpha 1,6 glycosidic bond, and alpha 1,6 glycosidic bond generally leads to branching, whereas alpha 1,4 and beta 1,4 glycosidic bonds leads to formation of straight chains of glucose. Cellulose has this beta 1,4 glycosidic bond. Okay. Is this clear to everyone? Let me know if you have any doubt. Which of the following nitrogenous base is a purine? Options are guanine, thymine, cytosine, and uracil. Which of the following is a purine? Guanine, thymine, cytosine, and uracil. Write your answers in the chat box. Okay. I can see most of you have answered option A, guanine. That is the correct answer. So you can see cytosine, thymine, and uracil are pyrimidine. Adenine and guanine are purines. So difference between pyrimidine and purine is the ring structure. So pyrimidine is a single ring structure having two nitrogens in the six-membered ring, and purine is a double ring structure with a six-membered ring fused with a five-membered ring, and there are nitrogens present inside the ring structure, and the base structure remains same in case of all three pyrimidines and in case of all two purines. The base purine ring structure is same. The difference is in the side chain. So like cytosine has this NH2 group and double bond O group here, whereas in thymine the double bond O group remains same as cytosine, but there's an additional double bond O group instead of the NH2 group, and an additional methyl group attached in the thymine. In uracil, the methyl group is removed. So if you remove the methyl group, if you demethylate the thymine, you will get uracil. In adenine and guanine, the purine base remains the same. In adenine, you have an NH2 group attached, and in guanine, instead of NH2, double bond O is attached in this place, and NH2 is attached in another location on this carbon. Okay. Moving on to the next question. Membrane proteins that transport substances from one side to the other are called. Membrane proteins that transport substances from one side to the other are called. Options are antiporters, symporters, uniporters, and none of these. Membrane proteins that transport substances from one side to the other are called antiporters, symporters, uniporters, none of these. Write answers in the chat box. I see two answers, two different answers, option C and option D. What about others? Others also answer. Let's have a tiebreaker. Okay, I have a completely different answer now. Option B. Read the options carefully. By the name itself, you can understand. So, something that is transporting a molecule from one side to the other. Okay, I have C, D, B. Three options. Mobini says all three A, B, C would be correct answer. What about others? Okay, I think the language of the question is confusing. Okay, the correct answer here is option C, uniporters. So basically the question like what the question meant was the membrane protein that can transport substances only in one direction from one side to the other. When it's written, it means that from one direction to the other and not the other way around. So in that case, the answer would be option C, uniporters. So uniporters by the name itself, it can only transport substances from one side to the other and not from this side to that side. Okay, so if it is transporting from extracellular to intracellular space, it can only transport in one direction. Whereas symporters by the name itself, you can see it has it transports one molecule along with a cofactor or a supporting molecule. So two molecules are always transported together from one side to the other, but two molecules are transported. Okay, antiporter, it is opposite of symporter. When one molecule is transported from one side to the other, the second molecule is transported from in the opposite direction. Okay, so the question meant to ask which of the transporter can transport only in one direction, basically. So the answer was option C. I can see that the language was slightly confusing, maybe. I hope all of you now understand
Yes, ma'am.
the answer. Yeah. Anyone has any doubt in this? Let me know if you have any doubt. Okay, so this is the last question of today's session. So anybody has any doubt in any of the questions discussed or any of the other questions in this topic? Anybody wants to ask anything? In the previous class, you guys were asking about the radioactivity question. I hope that is rectified on the portal now. The carbon dating question, is it rectified on the portal? Has anybody checked? Hello? You haven't checked it? I respond something. Have you checked it or not?
Hello, ma'am.
Yeah.
Ma'am,
Have
this
you?
is still showing 13 species.
It is still showing 13C. Okay.
Yes, ma'am.
Okay. I could see which this is the last question in the assignment list, right?
No, ma'am. It was question number 11.
Question number 11.
Yes, ma'am.
Okay. Okay. Just I did check that day. I think I checked the wrong question then. Just let me check. Week 6, right?
Yes, ma'am. B13 species is showing.
Question number 11. Oh, to the age of the dead plants and animals. Okay. Did anyone of you mail the NPTEL team regarding this? You can mail if they don't rectify it by the next week. I would suggest one of you at least write a mail to the NPTEL team and mention that this question is marked wrong. because according to me it should be C14, and we don't have the like we don't have the access to change these answers. So one of you has to mail the NPTEL team, because these questions and answers are uploaded by the instructors and the NPTEL team. So okay, other than that, is there any doubt in today's session or any of the questions regarding today's topics? Okay, if there are no questions, then I think I can end this session today. Thank you all for joining.
Thank you, ma'am.
Yeah, I'll see you next week. Bye bye.